why don't I just go ahead and introduce Aaron. Aaron's research is focused on how minerals interact with their environment and their living things. Aaron holds adjunct teaching and research faculty positions at USC, and he's also an affiliate research scientist at the Jet Propulsion Laboratory. In addition to his research, he also oversees the Gem and Mineral Hall at the Natural History Museum, and he's he's regularly putting together exhibitions and new displays in that space. So I'm going to hand it over to Aaron.
Hi, everybody. I think everybody's muted. You can't talk to me, sorry. But I am going to share my screen now so you can see my presentation. Everything's good? All right, cool. So my talk is, so I've been working a lot on with JPL and USC about origins of life and looking for life on other planets, and just how would you go about doing that kind of work. And it's, as you can imagine, it's fairly complicated, but it's all about exploration. And I found this interesting poster online. This was from the National Park Service. This was, exploration of Earth in 1920, 1918-ish, when the first National Park Rangers took residence at the park, and they were trying to attract people to go there. But now, like, these are the posters that are being made in 2020. Visit Enceladus, the moon of Saturn. This is like crazy how we're thinking about how we can explore and search for life and search for, just explore the solar system. Now in 2020 with commercial space flights and things like that, it's a completely different outlook on life and outlook on the world. But not only Enceladus, but there's posters of Titan. These are all NASA posters, Ceres, and Europa. And I'm going to be talking a little bit about Europa today. I really like this poster of Europa, so I just made it my backdrop for my whole entire presentation. But Europa it's got this ocean world on it. It's got this crust of ice, and there's the planetary ocean underneath it. We haven't really spent much time exploring that area yet because there hasn't been a dedicated mission to Europa. But come 2024 is when the Europa Clipper is supposed to launch, and it'll get to Jupiter in 2027. It'll take 3 years to get there. But there's a lot of exploring to do, and there has been tremendous amounts of exploring already. So it all started with the Viking mission in 1976 when Viking 1 and 2 landed on the surface of Mars. It was just a lander, not a rover. And their primary function was to conduct some biological experiments in the search for life. What they found is they found nothing. And I'll get into that in a second of why that was. But they returned amazing photographs back from the red planet. 20 years later is when Pathfinder landed. This was the first rover on Mars, and it didn't go very far, but it wasn't intended to go very far. It was just supposed to go off of its lander and just drove about a few meters, look at some rocks really up close, and make observations. So what I found is it found past evidence of water. There was rounded cobbles, there was like stream beds and things like that, ancient stream beds, minerals that look like they formed from water. And it also observed ice clouds in the atmosphere. So now we have evidence of possible past water on Mars and also current ice clouds forming in the atmosphere. That same year, Galileo was going out into space and made a pass by one of the moons of Jupiter called Europa. And it has a relatively young looking surface. You don't see a whole lot of craters on it. Like our moon is completely riddled with craters. And it is so because it has a really old surface on it. So when we look at planetary objects and there isn't any craters on it, this is an indication that this is a relatively young surface, probably less than 100 million years old. So this means that the whole entire crust of the moon is being recycled. This is very exciting. Then in 2003, Spirit and Opportunity landed, the twin rovers, and they did a huge amount of science. This was, just to put it really succinctly, is that this was the first mission that had compelling evidence of a habitable past on Mars, finding water-forming minerals, finding all kinds of amazing things. And so the race was on. 2005, this is the Cassini mission to Saturn, and this is a moon of Saturn called Enceladus, and you can see that these are geysers just shooting out from the moon. And the Cassini passed through some of these geysers and made some chemical measurements. And this is salty water. This is brine water coming out from Encelada's subsurface. So there's lots of salts, lots of water. Things are starting to get interesting. 2007, the Phoenix lander went to the poles of Mars. And you see that white stuff in the picture, that's ice. I mean, this is about as nail in the coffin as you get that there is water on Mars. This is near the poles, just in the subsurface. But it also found perchlorate, which is basically kind of a bleachy kind of chemical. And this is the reason why the Viking mission wasn't able to detect much in the way of organics, because the Viking mission, how it had its mass spectrometer is it heated up the chemicals, passed it through the mass spectrometer. But if you heat up organic chemicals in the presence of perchlorate, the organics just completely get destroyed. And so Viking mission from ground one was conducting kind of the wrong experiments, but it didn't know. It didn't know that there was perchlorate, and we didn't even know until 2007. Curiosity, this is the rover that takes tremendous amounts of selfies on the surface of Mars. That's all I've ever seen about on it on the internet. But it found huge amounts of mudstones. It is doing mineralogy on Mars, the first time ever. It was looking at chemical fingerprints. It found nitrogen, oxygen, phosphorus, and carbon. Found evidence of persistent water, liquid water on Mars. This is even more evidence mounting that there may be a habitable life on the planet. So the next mission is already launched. It's called Perseverance, the Mars 2020 mission, and it's going to land in a few months on the surface of Mars. And this is what it's going to be doing. We made this animation a little while ago. It's going to look for salt crystals in this environment. It's going to land in a crater that was an old lake, and was thought that this lake had water in it at one point in time. We see deltas and other river-type formations, but it has since evaporated. What happens on Earth is that when water evaporates, it leaves behind a mineral residue, and we call those salts. In this particular case, there's a lot of halite in that crater. But as these salts grow, they just grow in this stepwise fashion. And as they get larger and larger and larger, right in the little nooks and crannies of the salt crystal, liquid water gets trapped. This happens on Earth. And then the crystal just keeps growing and keeps growing and keeps growing and traps these little water pockets inside of the crystal. And they will stay liquid water for eternity. As long as the crystal is fine, that water will stay liquid. But if we peer down into those liquid pockets inside of the mineral, on Earth, we find lots of bacteria still trapped in there. And I'll show some examples of this, and who knows how long they can live. But the bacteria that on the surface of Mars and even on Earth is subjected to huge amounts of ultraviolet radiation. So on Earth, what they do is that they secrete out beta carotenes and other types of carotenes essentially as a sunscreen to protect themselves. So it's a chemical, it's a biologically produced chemical that will last for a long period of time. And then we can easily detect that without ever having to crack open the crystal structure using some laser spectroscopy. So here's an example of the rover we're driving around this ancient lake bed and just zapping salt crystals looking for biosignatures of life. So that's one of its driving focus. Again, it's going to take a huge number of instruments with it, but that's just one instrument that'll do it. So there's a huge interest in salts. And this is where I really focus my effort on is understanding salt crystals on Earth and potentially in other places in our solar system. Salt is just any kind of mineral that grows from evaporating liquids. We know there's a lot of salts. This was confirmed by the Phoenix rover. There's sodium sulfate salts. There's also hydrous salts that were detected from orbit on Mars. And the Jovian moons have brine volcanism of salt brine eruptions that are happening right now and happening all the time. So there's big interest for understanding salts. Salt itself, as we think of it, is just halite. This is the mineral that we put on our French fries. This is the stuff that we consume on a day-to-day basis. This particular specimen, the large one here on the screen, this was from Searles Lake just outside of Los Angeles, and it's pink because it's stained with all those beta carotenes. It is super, super rich in these beta carotenes. Very easy to detect with spectroscopy techniques. But on the right-hand side, I'll move that. I don't know if your videos are on the right-hand side, but I'll move it around. There is, on the top right, is a green halite crystal. This because it has copper chloride in it. And then there's a blue halite crystal in the middle. That's caused by radiation damage from gamma radiation from natural radioactive elements. And on the bottom is another orange one, but that's not orange because of beta carotenes. That's orange because of iron oxides, just rust that is staining the crystal. So there's lots of different ways these crystals can adopt their colors. There's other salts. You can go to the grocery store and buy halite, but you can also buy pickling salts. Epsomite and alum are also kinds of salt. This is a picture of epsomite. It's a naturally occurring salt. It's kind of rare, naturally occurring on Earth, but it's easily synthesized. And gypsum is also quite common. This is an hourglass gypsum. It's got lots of sand grains. And those sand grains and dirt particles inside of it can hold onto DNA for long periods of time. Mirabilite is not stable on the surface of the Earth. This is a crystal I grew from an aquarium. It was about, I don't know, about 4 or 5 inches long. And as soon as you take it out of the aquarium, it converts to thenardite instantaneously. So it is not stable for long periods of time on the surface of the Earth, but it might be stable on Mars and most definitely stable on the icy moons in the Jovian planets. So this is something that we can't really study too well on Earth in its natural habitat. So that's why we have to do it in the laboratory. So we grow crystals in the lab. This is one crystal I grew in the lab. This is from a brine that I collected from Cyril's Lake. And I just wanted to see, like, where does the beta carotene go? Where does the bacteria go? What happens as these crystals grow? So if we zoom in really close, you can see these lineations, these diagonal lines here. Those are all fluids that are trapped inside the crystal. If we zoom in even farther, you can start seeing the pink staining, the beta carotenes trapped inside of these fluids. If we zoom in even farther, we can take a look at the bacteria themselves that are inside of the crystal. And this is a fluid pocket. So you can see this rectangular square, and there's a mouse just following a bacteria floating around inside of these crystals. I've looked inside the collection of some of our old salt crystals that are probably 100 years old or so, and there's still bacteria just moving about. They've been in darkness for 100 years, haven't been pulled out. They are all over the place. We can look at other examples of this. There's another fluid inclusion over here that shows all kinds of bacteria just floating about in these crystals. So they are everywhere. And the nice thing about it is that once the lake evaporates, all that stuff is concentrated inside the salt crystals, so there can even be trace amounts of bacteria. And you'll still be able to find them inside the crystals. So there's a bacteria, there's a rod-shaped one, and there's mirabilite crystals at room temperature that are persisting in this crystal. This also tells me that the environment is really closed off, that even mirabilite can be persistent inside these fluid inclusions for eons, even at room temperature when they normally wouldn't be on the surface of the Earth. So how do we study these things? Well, we look at them using microscopes, but we also have to be pretty clever because we can't normally study things on the icy moon conditions, because around the equator of Europa, it's about -300 degrees Fahrenheit. So we have to build icy moon, what we call icy moon chambers. And this is one of them that's at the Jet Propulsion Laboratory that we built. It has salt crystals on the inside of it, and we just seal it up, cool it down with liquid nitrogen, and study how the salt crystals form, how they evaporate, how they crystallize, and how they melt at different temperatures. And we can also introduce cryophiles, which are bacteria that are cold loving. But we also go to the synchrotrons, which is a national laboratory. It's super high flux of X-rays. And the reason why we do that is that we can use ultrasonic levitation to hold liquid droplets in midair as we're studying them. And we can cool them down, we can heat them up, we can change the humidity, we can do all kinds of things that might mimic the surface conditions of some of these icy moons. The reason why we do this is an ultrasonic levitator here at the Advanced Photon Source in Chicago, is we also want to understand these bacteria are living in these liquids, but what is the liquid that they're living in? What is the molecular structure of that brine fluid? And how can that help us inform how these things persist, live, and eat, metabolize nutrients? So that's why we do those types of experiments. We also do CT scanning, which is looking inside the crystal in 3 dimensions without ever having opened up using X-rays. And we can see the different densities of fluids. So these are really carotenoid-rich fluids here at the top, the brighter colors. And we have fluids down here on the bottom left that are not rich in carotenoids. And we can easily calculate fluid volumes from that. And when we're in the field, this is my colleague, Scott Pearl from JPL, we can use digital holographic microscopes. These are microscopes that we can take into the mines or even put on a satellite and send it somewhere. It has no moving parts. It doesn't have any lenses. It just has a laser, a detector, and a beam splitter. and that's it. And they're extremely robust types of microscopes. The nice thing about this is you don't have to focus it, because the whole entire volume of fluid that we're probing is in focus. You just digitally focus the sample up and down, and you can follow bacteria as they're swimming around. This is something that was built there as well at JPL. So we have all these techniques that we can use to study life in its environment on Earth and possibly in other places. But what are we really looking for? So what do we need to be able to measure that tells us that life is somewhere else? So NASA's definition of what life is a self-sustaining chemical system capable of Darwinian evolution. That is a really well-crafted statement that leaves the door completely wide open to interpretation. Because, you know, if you have two rabbits, for example, if you have two rabbits sitting next to each other, you know the rabbits are living and they can reproduce. But what if one of them dies? Then that one rabbit can no longer reproduce. Is it still living according to this? It can't go through Darwinian evolution anymore, but we know it's a living creature just by common sense. But when we go to someplace else, we don't have that sort of information a priori. We have to really think hard about what is life. And I'm not going to get into this philosophical discussion. If you Google search Life in the Universe, the pandemic series, a colleague of mine, Charles Cockle, is an astrobiologist, and he talks about this, and it's really super interesting. It's about a 20-minute video. It's quite nice. But I will end it there. So what would be signs of life? Well, a sign of life would be if you went to another moon or planet and you saw something swimming around in an ocean. I'd say that's a pretty convincing sign that something is living. Chances of us doing that is pretty nil, because these icy moons, they have super thick crust. We're probably not going to get through them anytime soon. Will we find fossils? Possibly, but this has been a sticking point in astrobiology and planetary science, because there's a lot of these bacterial fossils can be formed by inorganic means. So if you find a fossil, that doesn't mean something was living at one point in time, because according to NASA, it's got to be a self-sustaining chemical system capable of Darwinian evolution. We know of fossils that live on Earth because we have a huge number of those fossils. We can trace back their lineage. Not so much when you go to another planet or moon. And should we search for molecules that produce forms of evolved chemical forms of evolved species, like the beta carotene is a chemical on Earth that evolved from bacterial evolution to help protect itself from ultraviolet radiation. Are those the chemicals that we should be looking for? And I'm going to say yes, because that's what I'm doing. But these are lots of different kinds of questions that we have to think about. I mean, is this the proper way to be doing it? Are there more intelligent ways to go about it? And it's the questions that I am constantly thinking about. But where do we start looking when we start doing this? The solar system is gigantic. It takes years and years and years to get anywhere. So you have to be very careful about what types of experiments that you do. So we start on Earth. We start looking at things on Earth. So again, going back to Cyril's Lake, here's what the lake looks like. This is just the field of view here is probably like 6 feet or so. There's a salt shelf on the left that's near the shore, and the water has kind of a salty crust on it, but it's pink in color because it's these carotenoid rich. Huge biological activity in that water. Where nothing else can live, these halobacteria survive. Here's the Great Salt Lake. Great Salt Lake is also pink. Lots of brine shrimp in there, and you know, birds live on those brine shrimp, and lots of other things going on. But this year, in January 2020, a new formation happened. This was really quite interesting, is that there's these salt things that started erupting in the middle of the lake. You can see them on the bottom right. You see these islands of white domes in the lake. These are these salt eruptions. This never happened before at the Great Salt Lake. And when you take a closer look at them on the top right picture, these are mirabilite crystals. These are mirabilite crystals that erupted from the deeper subsurface up to the top. They are not going to last for a long period of time. In fact, it only took a few days for them to be converted into thenardite, but it's so cold this time of year that they were able to persist for a long enough period of time for scientists to collect them. My colleague, Bonnie Baxter, who is the director of the Great Salt Lake Institute, she collected a bunch of these, but I haven't had a chance to look at them yet because I've been locked down since March. So hopefully she's going to send them to me soon so I can look at inside of the crystals and see if there's any bacteria. Because some of these eruptions are thought to be produced by biological activity. Here's the gateway to hell in Ethiopia. This is probably one of the most toxic and natural environments in the world. The water here has got a pH of about 0 on acidity level, which is even more acidic than stomach acid. is quite horrible. You would think that nothing can live here. The water is boiling. It's boiling acid and it's just salts everywhere. But you see these domes that erupt up. There's domes all over the place. These things are erupting because potentially of biological activity. And there's lots and lots of bacteria that are inside these domes that are trapped within the minerals. They're trapped within the mineral layers and they're trapped within the mineral crystals themselves. So what is this role between biological activity and mineralogical activity? And how do they work together in order to survive such insane environments? You can go down deep in the earth. This is about 1,000 feet below the surface of the earth, the giant crystal cave. These crystals are about 50,000 years old. They are in 136 Fahrenheit air. Getting down there is not pleasant. You can only spend about 10 minutes down there before your body begins to fail because it is so hot and so humid. But crystals that were collected here, and we have crystals on display at the museum from here, they contain bacteria, and these are about 50,000 years old. Penelope Boston, former director of NASA astrobiology, she was able to revive, her group was able to revive some of these bacteria and show that they were completely different than any bacteria that we had ever seen on the surface of the earth, further emphasizing their ancient origin. And we can go even further back in time and even further in the earth. This is me at the Balby mine. This is rocks that are 250 million years old. I'm almost a mile below the surface of the North Sea, about a mile, 2 miles east of the coast of England. And there's me hammering on some salt crystals looking for suitable crystals to bring back to the lab for analysis. And we took the digital holographic microscope down there and we're finding bacteria a mile below the surface of the earth. And so it's like, what are they living off of? How can they live like this? There's no sunlight. The water is horribly briny, super, super briny. What makes them tick? That was in March. That was right before the pandemic hit. And we just got the samples from England like 2 weeks ago. So, so things are moving slow. I don't have any answers for you yet. We're still doing the DNA sequencing of that work. So now we've got salt crystals. We have life detection, like what kind of chemicals we're looking for. And we have a potential place to go to, Europa. The Europa Clipper is going to launch in 2024. Hopefully, it's being built, it's funded. And what it's primarily going to be tasked with is going to fly around Jupiter. It's going to be in orbit in Jupiter, and it's going to make pass-bys around Europa. The radiation field around Jupiter is immense. You can't have any instruments that are in like constant orbit around Jupiter because it would only last for, maybe a month or so at most. So it's got to come in close to Europa and Jupiter, leave the radiation field for a while, and then make several passes. And it's going to come close to Europa's surface. It's going to come within 25 kilometers of the surface of the moon. Really kind of crazy close. It's going to look at ice thickness. Has ice penetrating radar on it. It's going to look at the surface eruptions and plume composition. So it's going to fly through some of these plumes, collect elemental analysis, chemical analysis to see what's there, and also have some spectrometers to see what the salt concentration, salt composition of these moons are, because we really don't know. We have no idea what sort of salts might be even there. We have some guesses, but we just don't know. Those measurements haven't been made. And so that's where we are. We're waiting for this mission to be built, but we're doing everything we can in preparation for it to try and predict what minerals might be there, and so that we can interpret the spectra and data a little bit easier. So that's where we're at.
Just one off the top of my head that we talked a lot about salts. I was wondering, what is the most common mineral in the solar system?
That's a tough question because we haven't measured, but that's a really valid question. I would say it's ice. I would say ice is the most common mineral. Ice is a mineral. It is naturally occurring. It is a crystalline substance. We see lots of comets made out of ice. The last mission to Pluto showed that it was just basically a giant ice ball out there. You have all these moons with large amounts of ice on them. So if I were to wager a guess, if I was a betting man, I would bet on ice as being the most abundant. I could very well be wrong, but we just don't know yet. Yes, Rob.
In a year when COVID is a part of our distant past and you can be doing whatever you want and you have all the funding you need, what would you be doing?
What would I be doing? I would be building my own satellite to send to Enceladus. to do mineralogy. That's what I would be doing. I think finding life on Mars would be pretty cool. It's going to be really hard, but I think these icy moons have a strong potential for harboring life because there's a lot of heat produced at these moons. So there's heat energy. There's potentially lots of nutrients as well. So if I were to continue to pursue this line of research, I have other projects going on too, but I think I would do that with infinite funds. I would not take myself. I would not go to Mars. That's a one-way trip.
Well, I heard Ed's interested in going to Mars now that he's out of school, so he can go. And does any of this relate to things you can share with a museum audience? Do you see any exhibits or anything that people who visit our museum could learn from all these?
I think so. I've been in talk with JPL. They're very interested in working with education group here at the museum. And it's how we go about exactly what do you bring? Do you bring a rover to the museum? Exactly how would that look like is pretty challenging. Instrumentation, but I think the story really needs to be a compelling one. And maybe getting some of these giant crystals, or maybe collecting all these brines and showing these different environments with help from the Jet Propulsion Laboratory. Or when JPL brings back Mars rocks, it would be pretty cool to bring some of those Mars rocks to the museum. There's some really interesting things that could be done. Any other questions? Anyway, I'll say Sue. Yeah,
Okay.
Sue's got her hand up first. She's unmuted.
Just wanting to relate it to your presentation is the Himalayan pink salt that you can buy in the market full of carotenoids.
No, they're not. That is from iron oxide. So there's no iron oxides.
Iron oxides.
Yeah.
Oh, really?
Yeah. So it's just rust inside.
So
of it.
you're eating rust if you use that.
Correct.
Okay. Thank you.
Yeah, no problem.
Interesting.
Yes, question.
When you talk about the bacteria that you're finding inside these samples, and you're not actually going in and taking the bacteria out of the samples, is that because there's the potential for these bacteria, we don't know what they are, what the consequences of releasing these bacteria would be, or it's just easier to study them inside their natural environment, for lack of a better term?
Yeah, there's a couple of reasons. I'm not so worried about the contamination issue, because once these bacteria touch even seawater is too fresh for these bacteria, and their cells will lyse. They'll just basically explode when they touch that. So even if it does get into the air, I don't even think they're pathogenic. So I don't think it's going to be a problem. The reason why I'm trying to do all this work in situ without ever having to open up the crystal is because that is probably the most related kind of work that NASA's interested in, because they're not going to be able to go to these other planets and moons and open up a crystal and start doing DNA sequencing, at least not yet anyway. They might be able to in the future, right away. And even if they bring them back to Earth, we want to be able to analyze them without any kind of contamination from earthly bacteria as well. So the more you can do on the crystal without having to get inside of it, the better the data is going to be, the better interpretations are. Eventually, yeah, we are going to have to crack open the crystals, and we are going to have to pull out the bacteria and sequence them just to figure out what makes them tick. But analyze them first and then do that second. And we are doing that. I don't do any DNA sequencing work. That's all JPL stuff. But yeah, those are two great questions for sure.
So when you say that a bacteria is 50,000 years old, it's not that particular bacteria is 50,000 years old because it's replicated and replicated and replicated. It's just that it might have started 50,000 years ago.
The interpretation is that they are indeed 50,000 years old because they go in the state of dormancy really quickly. So as soon as these halophiles get inside of a crystal, they're alive and they're kicking and screaming for about a month or two. And then they start off these rod-shaped ones, and as soon as they get trapped inside the crystals, they start to they transform. They start breaking up into these round cocci shapes. And I understand that as being they're trying to preserve as much energy as possible. They're shedding a lot of their flagellum, and they're just becoming these round spherical beads. They have huge amounts of RNA inside of them. And so they use that RNA to constantly repair the DNA that's there. They have more RNA than a lot of other living things. So there's ideas that they can persist for eons and eons and eons. And then finally, once the crystal dissolves and they get released out into the environment, then they can be reactivated again and go. Now that's for the 50,000-year-old one. Now the ones in Balbi, the 250 million-year-old bacteria, I do not currently believe that they are 250 million years old. I think the ecosystem that they're living in has been around and separated from the surface of the earth for 250 million years. And even then, I still question it. How can there not be any cracks that go down that far for 250 million years? So we're still doing a lot of work. I mean, such an ecosystem that's been separated from the surface of the earth for 250 million years old is going to require a mountain of evidence to really prove. Then we're slowly building that up. So there might be a couple scenarios to work with.
Thank you.
Doug.
The Chinese are on their way back from the moon.
Yes.
What do you think they're going to find?
I don't know. I think they went to the dark side of the moon to collect samples. So they could find water-bearing mineral phases on that side. They might find all kinds of interesting new minerals that have been cosmogenically been bombarded back there. It'll be interesting. They could just find the same stuff that we found from the Apollo missions too. It'll be interesting. Looking forward to the return for sure. Shauna.
Hi, Shauna.
Shauna, okay.
So I have a question about the bacteria that are, they're in these crystals for you don't know how long, right? And they're in torpor, but or they're hibernating. So what happens to the environment around the bacteria over time? Like, how do they change their environment? Because they're metabolizing, but it's like super slow, right?
That's exactly right. Exactly how they're metabolizing, we're not sure yet. There are magnesium reduction, oxidation processes that might be happening. Sulfur as well. There's huge amounts of sulfur in these fluids that they could be using for a food source. But the fluids themselves actually migrate through the crystals. So once the bacteria starts changing the chemistry, it actually freshens up the water a little bit on one side of the fluid. And then some of the crystal dissolves into that. And the fluid basically just starts marching around through the crystal over eons and eons and eons. You see evidence of that from the changing in shapes of fluid inclusions that we have. So some really, really old salt crystals often will have fluid inclusions that are not square anymore. They'll be rounded. So that's indicating that there's some kind of erosion happening on the inside of the crystals. But the process is so slow that it's really hard to measure exactly how that's happening. So that's just one hypothesis that's out there of how these things could do that. Changing of mineralogy. So if we see some mirabolite crystals or not mirabolite crystals, that can also be an indicator of what might be happening inside these fluids. But again, these are really long time scales we're talking about. So it's pretty tough. That's a great question. Yeah. I wish I knew more about that.
So can I ask one more? So how do you plan to date the bacteria? Are you going to look at like a molecular clock, or are you going to look at the environment, like within the bacteria, or within the salt crystal? Or how are you going to know if they're 250 million years old?
Yeah, well, the way we know that they're 250 million years, at least because of the deposit, the salt deposit is 250 million years old. That's been dated by radiometric methods using uranium lead and things like that, because there's natural radioactive materials within the salts as well. So the assumption is that the salt hasn't been changed at all since its original deposit. So you're getting the youngest possible age from those salt crystals. Now the younger stuff, like Cyril's Lake, we can go down about 150,000 years up to about 500,000 years at Cyril's Lake. And you can get each salt deposit that's along the way. And there's clay layers in between each salt deposit. So there shouldn't be any transmission of liquids between layer to layer to layer because the clay is impermeable. And so we're doing that work right now is trying to figure out, can we do the molecular dating? Can we look at evolutionary changes in these bacteria over time and get a sense of, you know, how old these things might be? If it turns out that the liquid water is just circling about in the rocks, then everything should be about the same age. Everything should be about the same molecular chemistry as well. So that's not my work, but I think I had one biology class in my life. But we're definitely talking about those things, like how do you date it, especially if you go to Mars, like how old can you figure out if those bacteria would be? Are they ancient or are they modern? It's a great question. Lauren.
Hey, sorry, a quick question. How do you pick these kinds of projects, and what else are you working on right now?
So yeah, it's a great question. So I'm a mineralogist and crystallographer, geochemist. So I mainly look at how minerals change in different environments. And so I've been working on this Mars project now for about, I don't know, maybe 10 years or so. And it really kicked off as soon as I came to LA, because that was 5 years ago I came to LA for the museum. And I'm so close to JPL that my collaboration there kind of exploded, and I've been doing a lot more work. But I do other things like rare earth element chemistry. I've done some consulting work for pharmaceutical companies, trying to cure diseases using minerals. So hyperkalemia, for example, we developed a mineral to treat hyperkalemia, to rescue people from hyperkalemic attacks using mineralogy and crystal chemistry. That's still ongoing work that I'm doing, but that paper should be published soon. It takes forever to publish these things. But yeah, this is just a large number of stuff. Also working with some local artists, and we're trying to clean up the Superfund sites in LA. So there's high levels of lead around Vernon. And so we're developing an arts installation / mineralogy way of cleaning the soils for the people that live in that area, so that they can bring down the lead toxicity to something more reasonable. So we're doing lots of work. And that's probably not even, I've got a webpage with a list of major projects that I'm working on, but the NASA one is a big one. Thanks. Good question. Doug, another question.
Anybody talking about trying to get under the ice and selling this?
Yeah, that's another project I'm working on. So we just got a discovery mission funded by NASA, and discovery missions are sort of an exploration money to try and do something. It doesn't mean it's going to get launched or get sent out in space, but we're building a drill. Basically what we're building a drill so that it can go to another planet or moon, drill down through some layer. And then as the fluids are coming up through the drill, we're going to use digital holographic microscope that'll be on board the drill and constantly look at the fluids that are coming through and looking for signs of life. Basically just looking for anything swimming around in kind of water. The nice thing about digital holographic microscopy is that it uses a laser. So we can do molecular spectroscopy at the same time as we're doing optical spectroscopy on these same things. So you try to JPL like multi-technique sort of instruments. But yeah, we're building those sorts of things right now. Great, that's a good question. Yeah.
Do you know how much energy it's going to take to get under?
I have no idea. The engineers at JPL are phenomenal, but not the batteries themselves are going to have to be withstanding the freezing temperature of the cold. I was just speaking of the kind of the opposite end of the spectrum. When I was on site at JPL, I was in the battery facilities laboratory, and they're building batteries to go to Venus. But the battery won't work until it actually lands on Venus when it's really hot. And so they're building these things to just fit the environments that's totally necessary for them to work. They're just magicians on what kinds of stuff they can build. It's amazing.
we're just about to wrap. I think if there aren't any more questions, I'm going to ask you to join me in thanking Aaron for another mind-blowing presentation. You never disappoint me, Aaron. I'm always fascinated, and I found that exceptionally so.
Thanks.
And it's really an honor to be working with you at the museum. I want to thank all of you for joining us today. I know there are lots of Zoom invitations out there, and we appreciate that you took the time to join us on this Sunday. I hope you guys stay safe and have happy holidays. If I don't see you before then, I do want to honestly say I miss every single one of you. I can't wait for the museum to open up again. This whole shutdown thing has been draining on all of us, I know, but there's light at the end of the tunnel. The vaccine's on the way. The museum will open again. And the reason that we're able to, be so optimistic and to sustain is because of the support of you guys. So I really want to share with you my heartfelt thanks and appreciation.